Now that we've covered how our body acquires, distributes, metabolizes, and eliminates lipids, let's talk about what our body can use lipids for other than as a fuel source. Think of this simply broken down into two main functional categories: the structural functions of lipids in phospholipids, sphingolipids, and cholesterol in all of our cells, and the signaling functions of lipids, mainly in the contribution of cholesterol for hormonal synthesis, but also certain unsaturated fatty acids for development of important cell signaling molecules. Let's start with probably the most simple but crucial role of lipids in the body, which is their role in maintaining the structure of all of our cells, most often in the form of phospholipids. But as we'll discuss, the lipid cell membrane of all of our cells can actually be diverse and differ not only in the polar head group or phospho component of the membrane, but also in the fatty acid tail, depending on our diet, which will come into play later. As we've said, lipids that we've brought to cells via chylomicrons and other lipoproteins are absolutely critical for animal cells, since they are the primary component of the cell membrane of all eukaryotic cells, which involve a lipid bilayer, specifically two layers of phospholipids, sphingolipids or cholesterol, or other analogs in other organisms such as ergosterol for fungus instead of cholesterol, which contain and protect the intracellular contents of all cells. Without this plasma membrane, we would be unable to regulate the environment within our cells needed to carry out critical cellular functions, and life simply would not exist in the way that we understand it. The plasma membrane's bulk is comprised mostly of phospholipids, but what are phospholipids exactly? They represent a large family of amphipathic lipid molecules, which comprise a polar head group, usually a glycerol molecule, bound to a phosphate group and then some additional R group, which faces the extracellular and intracellular environment, and a hydrophobic fatty acid hydrocarbon tail that we obtain from our diets, the fatty acids attached to triglycerides, or that we make ourselves through fatty acid synthesis, which face each other in this lipid bilayer, two layers oppositely facing with their tails pointing towards each other, creating this hydrophobic wall. Although there are many phospholipids, there are four main human plasma membrane phospholipids, although various fatty acid tails, four main molecules differing by their head group. Three of these four are glycerophospholipids. Take a look at the pictures to the right. The first picture, red square, identifies the glycerol backbone, i.e. this three-carbon molecule with three hydroxyl groups bound, one esterified to the fatty acid and another to a phosphate group, hence a glycerophospholipid. The fourth major human phospholipid is actually a sphingolipid, and more specifically sphingomyelin, which contains a slightly different sphingosine backbone rather than glycerol. See in the sphingomyelin structure to the right, actually a nitrogen atom attached to the central carbon of the three-carbon skeleton instead of a hydroxyl group. What we've already introduced here is an important biological concept that plasma membranes can differ in their biochemical composition, both in the composition of the fatty acids in their tails, aka the omega-3 fatty acid ratio versus monounsaturated fatty acids versus saturated fatty acids in their hydrophobic region, but also in their polar head group composition, aka the ratio of phosphatidylcholine to phosphatidylserine to more unique molecules like sphingomyelin or galactocerebroside, which may be more likely to have carbohydrate antigen moieties in their head group. This actually makes sense because when you think of a specialized tissue like nervous tissue, did we expect identical membrane composition to, say, for example, our esophageal mucosa? Sphingomyelin may be important specifically in myelinated nervous tissue, as well as galactocerebroside, which could at least theoretically be the site of pathology if membranes had abnormal compositions of specific phospho or sphingolipids. You may have also heard of the term ceramides, which is commonly advertised on skincare products, referring to a family of waxy lipid molecules composed of N-acetylsphingosine and a fatty acid. We have these variants of sphingolipids in many of our cells already, and we can create them de novo via hydrolysis of sphingomyelin. Ceramides have probably many cell-to-cell signaling roles in regulation of apoptosis and growth arrest, among others. Interestingly, they may have deleterious prognostic value since they have been linked in biopsy, if present in cells, certain ceramides like C16O or C18O for major cardiac adverse events and possibly insulin resistance. In skincare products, however, they are advertised to assist in the waterproof barrier of the skin. They are probably important like many phospholipid components of the cell membrane, good with some amount, bad with too much. Some of the biological differences that are exerted by this plasma membrane heterogeneity have been described. For example, the type of fatty acid tail seems to be important. Specifically, saturated fatty acid tails with no double bonds seem to be more stable and immobile at room temperature, with more stacking van der Waals forces between them, just like with saturated fats being solid at room temperature, whereas unsaturated fatty acids, which are liquid at room temperature, seem to allow for a more fluid membrane, probably because they force kinks and spaces between individual tails in the hydrophobic region. In addition, cholesterol, a major plasma membrane constituent in all animal cells, provides a buffer-like function, since in cold temperatures, when the cell membrane becomes more rigid, it allows more fluidity by creating physical space in the membrane, whereas in hot temperatures with higher kinetic energy, the cholesterol limits the amount of movement of nearby phospholipids, preventing the loss of shape. So in this way, fatty acid saturation and cholesterol composition are the two major players affecting membrane fluidity and stability. The identity of the polar head group is a less well-described factor in tissue and cellular differences, although not fully elucidated, it is at least reasonable to conceptualize that there are probably important intercellular and cell communication differences, since these polar head groups are environment-facing and therefore, as a part of the cellular lipid raft, may contribute to specialized tissue function. As well, some specific phospholipids, for example phosphatidylinositol, are known signaling molecules and, as we'll discuss, play specific signaling roles in the body. Therefore, it's clear that cells with different membrane compositions have different functions, and this may be more so dictated by its membrane polar head and backbone composition. Now, we've discussed the overall structure of our plasma membrane phospholipid bilayer as opposed to a micelle or bicell, which have one layer and thus all contents must be lipophilic. This membrane, including its phospholipid tails and head groups, is actually more of a dynamic structure, constantly shifting and changing, probably as individual lipids are accessed for other purposes or the cell performs functions and communicates with other cells. This shifting dynamic has been described as the fluid mosaic model of the plasma membrane, specifically describing movements of the layers. And so, for example, the outer and inner layer may flip and exchange places. We need ATP for this major change, typically occurring slowly, but this could be sped up or catalyzed by enzymes called flopases, which move the inner layer to the outer, or flipases, which move the outer to the inner. An enzyme called a scramblase can flip both of the layers simultaneously, free of charge, no ATP, if done in one swift motion. A less energy-intensive movement is, of course, lateral movement of phospholipids in the same layer. We call that transverse movement, and this occurs spontaneously. It's also important to review types of ways that biomolecules can traverse this membrane. Remember that the simplest way of motion through a membrane is simple diffusion, occurring best paracellularly for small ions or water, and through the membrane directly, actually diffusing directly through these phospholipids for very lipophilic molecules. Remember, in organic chemistry, we learned that very lipophilic drugs like those with a log P of greater than 2 can do this spontaneously. Also remember that proteins may help with passive transport, as in facilitated diffusion, so for example protein channels, or via osmosis with aquaporins, but simple diffusion requires no plasma membrane elements whatsoever. Primary active transport is a transport method, of course, that involves pushing something against its gradient and therefore requires some energy input. So for example, sodium-potassium ATPases, which require ATP to pump sodium into the lumen against its gradient. Or in secondary active transport, where pushing something else up its gradient can be accomplished by symporting or antiporting it with something else down its respective gradient, which itself does not require ATP. Lastly, we introduced the idea of lipid rafts, which float on the surface of a plasma membrane and primarily consist of cholesterol and other external constituents like carbohydrate antigens and proteins freely floating along the surface of the PM. Carbohydrates may be signaling, for example, ABO blood type antigens, and proteins can be enzymatic and have specific functions like the heads of or external domains of receptors like GPCRs or voltage-gated ligand channels. At least conceivably, the composition of lipid rafts could be important if there was some sort of pathology expected in cell-to-cell signaling in a specific tissue. That covers the surface of the plasma membrane, but what about the part of the plasma membrane which comes into contact with the membrane of other cells? We call these plasma membrane junctions, and different biological makeup of these junctions can dictate what can occur in this critical space. Conceivably, there are tissues in which we would want absolutely no movement in between them. So for example, waterproof barriers such as in epithelial cells. We call these junctions tight junctions, and they hold the two adjacent plasma membranes together using proteins called claudins, which are branching protein networks tightly sealing the junction. Cells without tight junctions can typically diffuse water or small ions between them. There are other types of cells where we would want continuity between the cytoplasm of one cell and an adjacent cell, which would help with direct communication of ions and water from one cell to the other. And this is facilitated by small donut-shaped tubes between one cytoplasm to the other called connexons, consisting of 6 connexin membrane proteins forming something called a gap junction. Gap junctions are critical for cells that need to communicate information quickly from one cell to the next. So for example, an electrical impulse guided by the flow of electrolytes like sodium or potassium, and thus, as you can imagine, are present in cardiac muscle. So the heart electrically can fire in unison. We call these intercalated discs in the heart muscle, as well as in nervous tissue. The last type of junction are desmosomes, which function to hold two cells together so they don't rip apart with stretch. So obviously, they are important for tissue that does stretch, such as connective tissue and muscular tissue in particular. So for example, intercalated discs in heart cells use both gap junctions to pass electrical impulses, but also desmosomes to prevent myocardial ripping with stretch. Desmosomes are composed of proteins called cadherins, which help them adhere, whereas claudins seal a tight junction, as well as an abundance of intermediate filaments connecting the two cells. As a clinical correlate related to desmosomes, we can imagine that failure of desmosome protein structure to remain intact could threaten the integrity of connective tissue, and indeed that is exactly what we see with a disease called Pemphigus Vulgaris, an autoimmune disease where IgG antibodies are created targeting the desmoglians, specifically the DG3 cadherin proteins, critical to the desmosome in the skin and mucous membranes, leading to loss of cell-to-cell adhesion under stretch, causing the skin to peel and rub off in a problem we call acantholysis. This clinically presents in patients with painful erosions to the skin and oral mucosa, possibly with flaccid bullae in the skin, and then subsequent ulceration and erosion with rupture. These are typically generalized, as you'd expect, with the nature of the problem, and can unfortunately involve mucous membranes of the conjunctiva, vagina, and nasal mucosa as well. The physical exam finding of Nikolsky sign describes epidermis that is easily removed laterally with rubbing due to the aforementioned acantholysis. And diagnosis is clinical, but then also involves histopathology, looking for findings of acantholysis and possibly serology if able to identify a culprit IgG molecule in the dermis. Treatment requires corticosteroid administration, which assists with most autoimmune diseases, and reduction in T and B cell function, but possibly other immunosuppressant medications, IVIG plus minus plasmapheresis to remove circulating antibodies. Pemphigus vulgaris might be more common in patients of Mediterranean descent, although few familial cases have been reported.